Hi, my name is Elyse Geoffroy. I am a master's student at Loyola University, Chicago, and today I'll be discussing inferring gene-trait associations with diverse population gene expression prediction models. In the past decade or so, genome-wide association studies, or GWAS, have grown in popularity, but have primarily focused on European individuals. However, these results cannot be accurately applied to non-European individuals due to differences in genetic architecture. As of last summer, 78% of individuals in the GWAS catalog studies were of European ancestries, while only 2.4% were of African ancestries. To combat this disparity, we use non-European GWAS results to perform transcriptome-wide association studies, or TWAS, to find genes associated with complex traits. Wojcik et al. 2019 is the largest GWAS study of non-European individuals, using approximately 50,000 individuals from the Population Architecture Using Genomics and Epidemiology, or PAGE study. These individuals are mostly Hispanic and African American individuals, and the researchers looked at 28 clinical and behavioral phenotypes, ranging from type 2 diabetes to the amount of coffee one consumes per day. Using these PAGE GWAS summary statistics and two of the multi-ethnic study of atherosclerosis, or MESA models, specifically the African American and Hispanic combined model, because it most closely matches the genetic architecture of the PAGE individuals, and the European model because it is roughly the same size as the AFHE model and provides contrast against the PAGE summary statistics. So using these two data sets, we perform SPredictScan. SPredictScan is an extension on the TWAS method PredictScan, which tests predicted gene expression levels for association with complex traits. In PredictScan, gene expression levels are predicted from genotypes, whereas summary PredictScan, or SPredictScan, which we use, uses GWAS summary statistics to generate gene trait association results without the need for the individual genotypes. So after performing SPredictScan, we then identify the number of significant genes for each phenotype using the Bonferroni threshold of p is less than 0.05 divided by the number of genes in the MESA model. Using this, we identified 151 and 90 genome-wide significant gene trait pairs using the AFHE and European models respectively. Out of the 28 phenotypes tested, 13 came back with significant hits. The white blood cell count phenotype had the most significant hits. However, this makes sense as the MESA models are built on the monocyte data of those individuals. Since we know that linkage disequilibrium and the co-regulation of genes can affect our SPredictScan results, resulting in many false positives, we colocalize our results, which looks at the EQTL and GWAS signals and identifies if they overlap, which suggests that these are more likely to be causal genes than genes that are not colocalized. We used two softwares, Coloc and Locus Compare. With Coloc, we identified 32 gene trait pairs in AFHE with colocalized signals and 20 in the European model. 6 were significant in both AFHE and the European MESA model. Locus Compare helps us to visualize the GWAS and EQTL signals to determine if they overlap. If a SNP or a cluster of SNPs meets the threshold of the GWAS and EQTL p-values being less than 1 times e to the negative 6th, we can label the gene as colocalized. We found 69 gene trait pairs in AFHE that were colocalized and 65 with the European MESA models. When looking at the overlapping colocalized genes between Coloc and Locus Compare, we then look to see if they replicated in PhenomeScan, which is a new resource that has the S-Multiscan cross-tissue TWAS analysis from the GTEx version 8 models with various European GWAS summary statistics. We found that 10 of our genes that overlapped in Coloc and Locus Compare replicated in PhenomeScan, and they are listed here. So finally, when applying AFHE and European MESA models to the PAGE GWAS summary statistics, we found more significant genes in SPredictScan and more genes that had colocalized signal and replicated in PhenomeScan in AFHE than the European MESA model. This suggests that when performing a TWAS, you should use a transcriptome model that most closely represents the genetic architecture of the original GWAS study. We also found 3 gene trait pairs that were not found in the GWAS catalog and may present new biology. These include ISCA2 and SETD9 for height and GPR84 for platelet count. I'd like to thank Dr. Wheeler for her mentorship and guidance. I'd also like to thank my fellow Wheeler Lab members for their support and collaboration, and Loyola for supporting this project through the Mulcahy Scholars Program. Thank you for listening.